Great, so our final keynote presentation this afternoon will be by Dr. Danny Singh. Dr. Singh is a professor of agronomy and a soybean breeder at Iowa State University. Upon completion of his PhD from the University of Guelph in 2007, Dr. Singh worked as a durum breeder with Agriculture and Agri-Food Canada before joining ISU in 2013. In his career, he's helped develop more than 75 varieties and germplasm lines with wide uptake by farmers in production fields. And he's published more than 150 research articles on topics related to plant breeding, genetics, phenomics, and the utilization of machine learning for plant breeding. And he also recently authored the textbook, Plant Breeding and Cultivar Development. I had the pleasure of visiting Iowa State University in 2019 for their workshop on machine learning for cyber agricultural systems, and I was thoroughly impressed by Danny's team and the integration of their plant science and breeding teams with their engineering teams there. So we're just tremendously thrilled that Danny was able to join us today, and I will let's welcome him, please.
What an exciting day, and it's fitting with the topic that I have. Thank you, Ian, for inviting me. It's always a pleasure to be here in Saskatchewan. I have some roots here. It has a special place in my heart, and it's good to see some familiar faces, and then just so much excitement with the kinds of topics that have been presented. What a grand success PERC has been. I've modified the title a little bit because it's good being the last speaker of the day, because you've talked about lots of different topics today. You've talked about different approaches. So I'm going to bring in my perspective with the team that I work in terms of the different disciplines as they come together, different approaches that come together. And I'm going to try and make a point about the diversity of people and where do we get that team formation that can lead to us being able to handle these complex challenges that are coming. So it's exciting with the technology, but it's also, we live in a very complex time. This presentation, there are 3 sections. In the first one is what do we do? There's only 1 slide on it. In the second section is how do we do it? And there are lots of slides, and I'll explain what the format is. And at the end, I'm going to conclude with the how. Who are the people that are engaged, people that are working in solving the problems, and who are the people that are providing the inputs for us to come up with those solutions. So we are a plant breeding group, as Ian talked about. We work with a lot of different disciplines, so it's a large team. I'll explain that team later on. But from a plant breeding perspective, a lot of things that we do in plant breeding relate to cultivar development. Those are the products, outputs that farmers grow. We also have to keep in mind that how do they fit in the crop production system? Because plant breeding and crop production system goes hand in hand. So thinking about those things, I've put, let me go back here. So those of you who are not in plant breeding domain, on your left side is a plot of soybean field. These are varieties. There are thousands of different varieties that we test each year. There are different stages, so that's the video that's showing. On the right side is a plot of millets. That's a crop that we have recently gotten ourselves into. It's a very hardy crop in terms of both heat and drought tolerance, so it's of interest to us. In the middle there, and again probably in the back you cannot see it, but the images in the back, they relate to our work in heat stress, drought stress. And I'm going to talk about it, so I don't need to explain it now. At the bottom of the middle, there is a drone-based application of nutrients, this abiotic stress, and how do we utilize aerial imagery. I'm not going to talk about that specific project. I'm not going to talk about diseases and nutrient stress as much today, but later on, if you have questions, please feel free to come and talk to me. So now we'll get to the crux of the presentation. In this next set of slides, there are going to be sort of 4 sections of this particular theme. In the first one, I've put some icons and some titles. They may or may not be visible. And the first is the types of different platforms that we use, from ground robots to drones to satellites, going into a multi-modular and multi-platform phenotyping. And then I'm going to talk about some of the work that we do in terms of application. There's role of sensors. How do we utilize the information that comes from sensors? And then I'm going to go into some of the topics that we are sort of new into. Ian wanted me to talk about our recently concluded work and also some of the work that we are doing and is not published yet. So I'm going to talk about some things that we are excited about. And finally, I'm going to finish with where we can have better representation and some of the initiative that we have taken in our group. So in the very first topic, ground robots, so let me back up. We approach phenotyping and tools that we need to utilize this, in terms of phenotyping. If there are aspects that we can see, and traditionally that we have used just visual assessment, what can we do to obtain those sets of data? Essentially, there is a component, there talks about the genomic component. I'm not going to talk about it. The solutions I'm talking about are related to the phenotyping, things that were done visually or through use of machine. How can we make things better? The first slide here in the video, you'll see is a ground robot. Right now, those of you who work in field crops, and those of you who have talked to plant breeders, we always say machine is the best, phenotyper. Combine, you get your machine, you get your yield, you know if it's going to be a keep or a discard. That's great, but as we are thinking of, the other aspects of plant breeding, we need to be thinking about how can we do things faster, how can we do more with less amount of time. These robots with the right camera, and these are just simple cameras, and that's what you can see in here is what the camera is looking at. Can we use a ground robot that can go in between these soybean rows and determine how many pods are there? These pods are the reproductive organs in the plant. So instead of a combine taking the yield and telling us, what is the yield on a per area, can we have robots to do it? Now, why is that useful for us? Combines, we have to travel, we have to go there. There's machine cost, there is depreciation cost, there is fuel cost, there's personnel that are involved. Can we use robots to do it? And in fact, we were able to show that yes, we can do it. This is the workflow. This is the workflow, the imaging, pod data annotation. Pods were annotated. We collected pods, we annotated it. We used an ML pipeline for the pod detection, and then we were able to do pod estimation. This video here, again, it's small. They're actually boxes that are shown. So this is an ML program. You can do it in real time. You can do it after the fact. So the robot is going and it's detecting the plants. It's tracking the plants, but it's also putting the confidence boxes about, this is a pod. What we have shown is that this works with enough accuracy that we can do our keeps and discards. We may not have, perfect accuracy and precision in terms of this is the exact yield, but in terms of the ranking, we can do it. So this saves us a lot of time. It makes our lives of our team members effective. And what does it come down to is that if we can get these tasks done with these machines and these ML tools, it gives us more creativity. It gives us time to do creative things. The second example here, so we have done work with yield estimation with drones and other things. I'm taking a different view of utilization of drone. This one is estimating maturity. It takes a group of anywhere from 10 to 15 of our team members to go into the field and estimate or visual estimation of what the maturity is. We are talking about a month of work by so many human hours. That's a lot of time for people to be doing something that could be done in an automated manner. So I hope you get the gist of what I'm trying to say. Anytime there is a task that can be done easier by a machine, that's where we are looking at investing our time to solve that problem. So in this case, these are same genotype, different maturities, that stages in the development of that plant. This is a video of the field where repeated aerial flights are taken. It starts with the earliest expression of that maturity, and we take repeated flights. A multi-view CNN model is used, and what that does is same particular plot that has multiple different instances of that being imaged, it is able to generate the relative maturity. What does that mean? That means 1 month of work, can be completed in 8 hours of that drone time that, for that field. We're talking about thousands of plots. In our program, we generally have about 100 acres of experiments that we grow and manage. So this is a big time saving for us. And hopefully it is also time saving for everything that we do. More recently, we have tried to make all of our data that we have and also the models on our GitHub page so that can be used by the community as well. So the next example I'm going to give, so I moved from the ground robot to drones. I'm going to talk about satellites. And I think in the audience, I don't know your name, but you asked a question about satellites. So satellites have come a long ways. I'm going to give a story of a farmer because it connects to the who towards the end. So this particular section that you see, and I'm not going to talk about why are satellites useful. We all know that if we can get satellite with the kind of work that we are expecting ground robots or drones to do, and a satellite can do it the same types of outputs, just imagine the time saved, all of those, okay? So there was a farmer colleague, and they know about the kind of work we do, and that farmer colleague asked that I had this problem of fall armyworm. Fall armyworm is an insect that comes from the south in the US, and it comes, even to the Midwest. When it comes to your field, it decimates the crop. This particular farmer, this is after the fact, asked that I scouted my field on a Friday, and I did not notice the instance of fall armyworm. But when I came back Monday, my crop was decimated, was destroyed. This was an alfalfa field. And the farmer colleague asked, could you, like the technologies that you work with, could you have done something about it? So my colleague, Dr. Ardy, and my PhD student, Dr. Matt Carroll now, just defended last week, they set out, took a look at the satellite feed that we were using at that time. And what they could detect was, so this is the alfalfa field of our colleague. There is instance that is forming, and these are different dates, and you can't see it, but you see the color change that is happening. If the farmer had access to this information, they could see or sense that where was the damage starting. They had two options at that time. They could have decided to mitigate, control the insect, or they could have baled their alfalfa field. They did not have to wait, a couple of extra days. They would probably have been fine. This tells us the importance of the kinds of technologies that we work and how it can impact hundreds of thousands of, decisions that a farmer makes. In our team, we are motivated that whatever we do has to be applicable for farmers and farming. So the problems that we take have to have some, connection that it ends up helping the people that we are trying to serve. So this is a powerful case for us because it shows that, with the technologies that we have, we can make it, usable and hopefully eventually accessible to farmers. We have moved to another satellite feed that is at 0.3 meters square per pixel. That means we can do plot level information. This farmer field was not at 0.3 meters square, but we could still detect, where the insect had started. So we have moved from the ground robot to the drone to the satellite. And again, the impact you can understand. And I should have, it's cutting out the people at the top who are working, the student who are working. And the layout of the slide is motivation, methods, analysis, results, and impact. This fourth example I'll give in this section, it's utilizing soil information. So we're not soil scientists, but soil is so critical. And it's just not thought of by plant scientists, but it is fundamental to what we do. Those of you who work in field studies, spatial adjustments is something that plant breeders utilize extensively because there's a lot of variation, and we want to be able to compare apples to apples. But that means that we have to be able to adjust for these differences that exist in the field. Right now, the state of the art is you look at how the neighbors are. So if my neighbor is doing worse, that means that, okay, we both need to be able to adjust our performance. We have not taken into account the soil. Now with soil mapping that happens, the advances that have happened in soil mapping and the advances that ML has done in terms of getting soil maps, we now soil map all of our trials. Using those soil maps, we can get to plot scale, features of the soil, these different soil parameters. So what we attempted to do here is utilize soil features in our spatial adjustment. So we are trying to understand, we can make the adjustment, but we are using extreme gradient boost to bring in the soil features on per plot basis. And we are doing our spatial adjustment, and we used 3 different ways of analysis to make sure that the improvements that we get from an ML-based incorporating these soil features, is it truly making an improvement in our spatial adjustment? And that's what the results here are showing. So if you're interested in the adjustment, if you can see in the back or maybe those in the front, you can see a lot of variation, but after adjustment, it homogenizes and you're able to compare apples to apples. In terms of the model effectiveness, this is showing that the XGB model has a lot of improvement over the baseline, and this is the state of the art that a lot of programs use. And there are different metrics that we use to be certain, because it's hard to track in plant breeding. It is you discard and you don't know what it truly means once you've discarded. So we have to go with what has been selected, and it seems like this method allows us to make better adjustment for these spatial autocorrelations. But more importantly, what it also means is that we have soil features that are telling us the contribution to these yield and also to the adjustment. So using Shapley values, we can take a look and we can determine that which feature is important in that adjustment and also in the estimate. Why is that important for us? So it's good that we could do these adjustments, but now if we want to make some selections for nutrient stress, and we know that this is the area of the soil that there is a nutrient stress, we have the explainability of utilizing that particular information. I'll move to some of the applications of use of different sensors. And of course, those of you who have heard me talk about, I'm very passionate about roots. I used to call it a carrier killer, but I love it. Being tenured, it makes you a little bit more brave. So I'm going to continue working on roots. So these are the projects that I've picked. So we work on a lot of different projects. I've picked a few projects which I thought were relevant to the topic that you have today. So I don't need to tell you, you've heard drought is, it's an incredible problem that is coming together. I have to also mention that it's not just drought, drought and flooding, it's the variability and extreme events that are the issue. But let's break it into smaller parts and then build the car together. So in this case is a drought nursery. This is our drought nursery that we established a few years ago. We can screen for couple of thousand different accessions to look at the response of water stress. It's a really good nursery for us. So we're moving from visual assessment, because visual assessment of drought, there is an inter and intra-rater variability, and we want to avoid both. We want to have no bias in our assessment, and we want to be able to, when we call something drought tolerant, it truly is. So what we are doing here is we are using multiple different sensor data, because we are trying to bring the different modalities to come up with better prediction. So if we use multiple different modalities, so let's say RGB, multispectral, hyperspectral, thermal, whatever different those modalities is, those modalities, the training of those modalities together can inform one of those modalities to do better. So RGB alone can do better because it was trained on multiple different modalities. That's sort of the premise. And then the second aspect is that you can actually have better prediction, earlier prediction than you would otherwise have. So that's what this particular project is. We've screened over 3 years, hundreds of different accessions replicated. We have lots of information that is informing our breeding process, but we are also advancing the ML component of integrating different data streams to be able to utilize that information for our breeding for drought stress. You can't talk about drought and not talk about heat. They kind of go together. But again, let's parse them out first. We've done extensive research on heat stress, and there is good genetic information. There is good genetic variability that exists in the accessions, which is good for a plant breeder, which means that we can try and put the genes together to improve our heat tolerance. But the current work is primarily done in greenhouses. And those of you who work in greenhouses and in field, I hope you agree with me that whatever we find in the greenhouse, it's a roll of dice that it may or may not hold in the field. because field is just so much more complex. So it's very good in the beginning for us to be able to utilize our greenhouses to study for heat stress. We want to be able to move it into field. What we have done so far is we have taken a look at a lot of different traits, combining those different traits, observing that, what happens in that high heat versus optimal temperature, and what is the plant response, what is the best phenotypic indicator, phenotypic trait that we should be utilizing for heat stress. So we have done all of that, but what was missing was that we need to go take it to the field. Now, how do we in Iowa create a situation where we have, no heat and heat, next to each other? So diversity of people, right? They have capstone courses. These are final year courses in engineering. There's a capstone team that we have worked in the last 6, 7, 8 years. So we engage these capstone students. These are final year mechanical engineers who are looking for new problems. We propose this problem to create a mobile, field deployable, semi-autonomous heat chamber. It can be set up in an hour, it can be disassembled in an hour, and during, between the time that it is there in the field, we can increase the temperature up to 4 degrees higher. We chose these degrees because we want to be able to see what's going to come in 10 years, 20 years, 30 years, 40 years. In plant breeding, each cycle can be anywhere from 5 to 10 years, depending on the crop. Maybe it can be more. So whenever you're thinking of developing cultivars, we got to be thinking about, what is it to be expected in 5, 10, 15, 20, and so many years. So this is an example of a heat chamber that we have utilized. It has received a lot of attention. That makes the student excited because they are working on a project that gets featured in press, media, YouTube, radio, television interviews. So students are very excited about it, and so are we, because it just shows the partnership that you can have with a team of undergraduate students, not from agriculture, working on, solving some of these complex problems. So I told you I love roots, and they're complex, and they're very, very difficult to work with, extremely difficult to work with. We currently do not have any good system that we can do repeated measurements of the same root in the field, at the scale that we work. So almost everything that I've talked about, our scale is generally several hundred to thousands of accessions at a time. We generally don't work with, few genotypes. So that limits what we can do. So one of the student here, so this is a plant science, partnership with an engineer. They developed this computer vision based, root phenotyping system that can, you take the image. This is in a blue paper. Again, it's not in the field yet, so it's in the blue paper. You take the image, and it gives out lots of different traits. You can see, the difference in the traits that exist, and that's very useful because using this information, instead of looking at individual traits are great, but sometimes it's the integration of traits. You're looking at categories because those categories can, inform us about the type of responses that we are looking at. So here we proposed an informative root category or type, and these are different categories that, have features that people have reported previously. So you can have a maximum type, you can have, a nutrient foraging, you can have deep, steep, cheap type, going deep. We heard about that earlier in the morning. You can have an umbrella type that is foraging, but is also trying to go deep. You can have a beard type that is just maximizing the density. You can have all these different categories. So we took this information and we made crosses because we knew what we have in our program, the kinds of root trait diversity that we have. So we're just, at this stage, just experimenting with it. Just bringing in new types of root system into our genetic base, because then we are linking it to the production, as we move forward. This one is nodulation, where you take the roots, but we forget about the nodulation. So how do we take the information from roots on the nodules? It's very difficult. If any one of us is given the task that you have to remove the nodules manually, none of us would take that job, I'm sure of it, okay? So we need that example that you need a tool that we can do the study of nodulation, and that's the work that Dr. Clayton Carley, who also defended last week, a PhD student in our group, came up with a partnership with Dr. Zaki Zubairi. with a platform Snap that does the assessment of nodulation. And what we were able to do is nodulation is not just total number of count, which is what we have done in the past. Because of the ML tool, we actually have the volume. We have where it is distributed, taproot, lateral root, which part of the root, how is it developed. We used a time series, and we determined that it's actually early in the time series that the nodulation is connected with the end season nitrogen. Up until this point, it is assumed that it is during the reproductive stage, but this is the first evidence that it actually is even earlier than that. So we are thinking of, the next move is to move from the nodulation part, having a look at both the carbon and the nitrogen, because the nodulation is the bacteria fixing the atmospheric nitrogen, providing it to the plant taking the carbon and giving it to the bacteria. And it saves a lot of time. I'm talking about 20x save of time. So these are three projects. I'll go quickly through them. Digital twin, there were some discussions earlier which I think digital twin could provide an answer. There are three components of a digital twin. There is a physical object, which is the plant. Could be a field. There is a second component, which is the replica of that physical object in a virtual representation or environment, which is the virtual space that you see on the figure. And then there is the connection, the data or the information exchange between them. Why this is important is that it allows people to run multiple complex simulations, which cannot be done on a plant. So you can use the biophysical models that provides the constraints, to the condition. ML model can go haywire. Sometimes they just go and make predictions that are not, going to be useful. So you constrain by giving the biophysical constraints to it through crop model, and you utilize the features of the crop model and features of the ML model to come up with ways of testing the responses in heat and drought. So this is an experiment that's done with lots of different sensors, looking at different heat temperatures as well as different water regimes. And trying again, the earlier example I gave, to combine fused data, these multi-modular data, to be able to run these simulations about what would the plant response be. This is useful for research scientists because it can also inform us the kinds of hypotheses we should find, but also about mechanisms of developing more heat and drought-tolerant varieties. And for farmers, because you can have mitigation, because you can see what is happening in your field, because your model is getting continuous feeding of real-time data from multiple sensors. This one is augmented and virtual reality. In the video that you will see, it may not be visible, but essentially what this is, a 3D point cloud. Again, it's downsample, it's in the Unity environment. This is a person wearing a headset. There are two, you'll see arrows. One is showing, when you click it, it shows what the expert rated for that particular stress. And on the other one is showing which particular canopy you're looking at. It's a great tool where a breeder or scientist cannot be in all places at all times. After the fact, you can go and check out what was the rating. What did that particular plant or that variety look like? You can also use it as a training tool, because you can get people that this is what the expert rated, how would you rate it? And it is quite, if you could see it, it is very clear. So this is, a way of advancing. Once we put the augmented reality in it, in terms of, all the other things surrounding, we're using 3D point cloud, but we have also used 360 camera. So there are multiple ways of doing this, and I think this is going to be exciting. Heat is, a big problem. We talked about it, but it also brings lots of different insects. So insects are complex. There's lots of variations. Same species can have different features. Different insects may look similar to each other. There's diverse background in the wild. Some are small, some are large. There are multiple stages. So we're projecting heat's going to increase, and we know that insects will also be increasing. So we need tools to be able to detect these insects. So we developed an insect detection app. This was a beta version of 150 species. Now it's moving to 2,500 species. Eventually it will be higher number. It can do invasive plant species. So invasive pest species. You can, things that are just new, it empowers. It's a citizen science to that extent that it empowers people that they can take a look. It also can be accompanied with IPM features about these insects, and it does a fairly good job. We have Recently, we have shown this app to more than about 10,000 people through different shows that we have done, and we have received positive feedback on this. And one of the things that I talked about earlier, there was someone else, I think you had talked about the actuation, the closing the loop. So we are into cyber agriculture systems. Ian was part of that machine learning for cyber agriculture system. And this is an example of, you have the sensing, which is where you're taking the data. You have the modeling, where the ML or any other kind of modeling happens. And you have the actuation, where a human or an object takes some mitigation step. So here, I'll quickly walk through it, and I'm coming to an end, Ian. So you have the camera that's going around, taking an image. The image sees and detects that the bug is there, and it can spray to control. This video is showing that dexterous robot that just moved. This is what it sees. It's recognizing the Japanese leaf beetle as the insect. With the nozzle, it can actually spray. This is closing the loop. You're sensing it, you're modeling it, you're able to detect it, you can do it in real time, and you're taking in some kind of action. You can imagine the possibilities. I'm just giving one example, the work we have done with insects, but you can imagine different possibilities that exist with this. I have to talk about this. I look in the audience and I see a lot of diversity, but I think we can do better. So this is a group of women in ag and AI. My colleague, Dr. Ardi, she has started. These are folks who are on the ag side and then on engineering side. And any of you who have an interest in joining this group, they work on a lot of different, like kindergarten all the way to K-12 and university and beyond. There's this is a good place, I think, good initiative, I should say, to work, together with this group. They are in Iowa State, but I think their aspirations are to go, even wider. And they've the images here. This is showing the work that is being done to farmers. This is, work showing through the Farm Progress Show to kindergartners. So I talked about the who, and there's only 2 slides left here. One of the things that I hope that you can, that I can convince you is each team has a lot of strength. There's no doubt about it. And I think you folks have figured out, how to do it. You've been successful. One of the things that I personally believe has helped me to grow and to be better is to work with farmers. And I'm not talking about that, we talk to them. I'm actually talking about having them involved in the visioning and having them involved in even the solutions. Incredible intelligence, incredible experience. Everything that we do generally has participation from these farmer colleagues. That has led to a $20 million grant, which is an AI Institute for Resilient Agriculture, that came from some of the discussions that had where 2 farmer colleagues were in the ML-CAS panel. They provided the problems that they deal with, and they talked about some of the solutions they believe we should be looking at. And I'm so thankful to the team that I'm a part of, that people were listening and they respected it. So this is an example where my colleague, Dr. Bhaskar Ganapati Subramanian, he's the PI for AI Institute. A colleague of mine, Dr. Shamik Sarkar, who's received the CPA Cyber Physical System NSF $7 million grant. It's incredibly rewarding to be a part of a team where people that we care about are actually embedded in the team itself. This is the team that I get a chance to work with. I'm very fortunate. Every day is just a fun day because I work with incredible people. These are the PIs that I work most closely with. The Soinomics team, we have been recognized with recently an award for our work together. And the team is growing. So these are some of the core members that when the team started, we have grown in our PI. And just a lot of, lots of different backgrounds of people that have helped us. They're the engines, they're the drivers of the innovation that we have been able to do. What I've done at least I've tried to do, is I talked about what do we do. I've tried to give some examples of how we are doing it, primarily ML-based phenotyping and the cyber agriculture systems. And then I've talked about the interdisciplinary team, the role of it, and then just the participation of farmers as we grow together. So these are the things that I tried to talk about. I won't repeat it. It's up there. I have to acknowledge the funding agencies that have enabled the work that we do, just different partnerships, and again, the support from producer groups in the US who have also helped along with the federal agencies. And this photograph is Dr. Matt Carroll, and I gotta acknowledge that's Matt and then Clayton here. They both graduated last week, so I'm very happy. They're gonna go on and do great things. Some of the things that I talked about, if you have an interest, Ian pointed out, some of those things I've talked about would be found in this book. And if you're interested in high throughput phenotyping in soybean, there's a book chapter that you can take a look at. Thank you so much for your attention.